Hello everyone, welcome to my channel Biochemistry by Dr. Tejas Shah. Today we will see about the metabolism of acidic amino acids and their amides. So let us first see some key points. Glutamic acid, glutamine, aspartic acid and asparagine all are non-essential and glucogenic amino acids. Glutamine is converted to glutamate and glutamate is further converted to alpha ketoglutarate and hence it is entered into the glucogenic pathway. Asparagine is converted to aspartate and it is formed oxaloacetate. It is also the TCA cycle intermediate and enters into the gluconeogenic pathway. Glutamate play a central role in amino acid metabolism because amino group of all the amino acids are converged to form glutamate through the transamination reaction. That is the first step in the catabolism of amino acids. So here the link with my previous video is with the catabolism of protein, and this is the link which I will also provide into the description so that it will help you to understand the central role of glutamine in amino acid metabolism. So let us first see the synthesis of glutamic acid. So it is synthesized by three ways. First is through the alpha ketoglutarate by the transamination reactions. So all alpha amino acids combined with the alpha ketoglutarate to form glutamic acid and corresponding alpha keto acid. Here pyridoxal phosphate acts as a coenzyme and the reaction is the transamination reaction. Also another reaction in which the glutamate is formed from the alpha ketoglutarate by the enzyme glutamate dehydrogenase, that is the reductive amination, where ammonia and reductive power is provided by the NADH to synthesize the glutamate. And third, it is derived from the glutamine by the enzyme glutaminase. Here the ammonia is removed and there is formation of the glutamate. So by these three ways the glutamate is synthesized. Glutamic acid is also formed during the metabolism of histidine, proline and arginine. Just quick overview. So from histidine through the intermediate glutamate, there is formation of the glutamic acid. Through the again glutamate is converted to glutamate semialdehyde, which is also converted to proline, and proline also converted back to the glutamate semialdehyde, and it is formed the glutamate. Glutamate semialdehyde, it is formed ornithine, and ornithine is synthesizing the arginine, and arginine enters into the urea cycle and form the urea and again ornithine. So these three, histidine, proline and arginine, in their metabolism will also form the glutamic acid. Now glutamic acid degradation. So as the transamination reaction is the reversible step, so glutamate combined with the oxaloacetate by the aspartate transaminase will form the alpha ketoglutarate and aspartic acid. This is the first reaction. Another is the glutamate dehydrogenase, the same reaction by which the glutamate is formed from the alpha ketoglutarate and ammonia. So glutamate again, if the NADH is available, then it is converted back to the alpha ketoglutarate and ammonia is liberated. Now some important metabolic functions of the glutamic acid. First is play a central role in the amino acid metabolism. Second, it is glucogenic through the formation of alpha ketoglutarate. Third important function is its synthesis. The N-acetyl glutamate, the positive modifier of carbamyl phosphate synthetase 1 that is located in the liver mitochondria and play important role in the urea cycle. Fourth is gamma carboxyglutamic acid of the prothrombin. So prothrombin having a glutamic acid in which the gamma carbon or at the gamma carbon carboxy group is added, and this addition is required the vitamin K as a coenzyme. So vitamin K play a very important role in this reaction. So it is helpful in the clotting process. Next function is the synthesis of the glutathione. Okay, And this is the link in which I have explained the glutathione in detail with its bio or important functions. So I will also share this link in the description also. Now next function is the synthesis of the folic acid. Here you can see the structure of the folic acid. Another name is the tyrol glutamic acid. So here you can see the glutamic acid molecules are incorporated to form the folic acid. Next function is glutamate acts as an excitatory neurotransmitter. How? So glutamate stimulates the N-methyl D-aspartate receptors or reacts with these receptors, which leads to open the calcium channels, which stimulates the nitric oxide synthase. So there is formation of the nitric oxide, and nitric oxide stimulate the guanylyl cyclase enzyme, which converts GTP to form cyclic GMP. So increased cyclic GMP level will lead to neuronal excitation. So that is how the glutamate acts as an excitatory neurotransmitter. Next function is synthesis of the gamma amino butyric acid, that is called GABA. So if we see the steps, the glutamic acid upon decarboxylation reaction where the pyridoxal phosphate vitamin B6 acts as a coenzyme, so there is formation of the gamma amino butyric acid. Now again this GABA upon oxidation by the GABA oxidase will form succinic semialdehyde, which further oxidize to form succinic acid and enters into the TCA cycle. Further it will form the oxaloacetate and further it will form the glutamic acid. So this reaction from GABA to again form the glutamic acid is called the GABA shunt, which is very important reaction. Now the importance of the GABA here. The gamma aminobutyric acid is a major inhibitory neurotransmitter. Now important point to note it here is glutamate acts as an excitatory neurotransmitter while its derivatives, that is the GABA aminobutyric acid, acts as an inhibitory neurotransmitter. So binding of the GABA to its receptor increases the chloride conductance from the outside to inside the neuron, hyperpolarization, and which decreases the nerve excitability. So that is how the GABA acts as an inhibitory neurotransmitter. Now important clinical point here, if you see this reaction, glutamic acid's conversion to the GABA requires pyridoxal phosphate as a coenzyme. So if this pyridoxine is deficient, that will lead to, or that will remove the inhibition and that will lead to the conversions. So sodium valproate, which mainly inhibits the GABA oxidase, and that's why the GABA level is maintained. So the convulsions or epileptic seizures can be controlled by this drug, sodium valproate. Now there is link between the GABA and the Huntington's disease, that is the neurological disorders where there is degeneration of the GABAergic neurons, which leads to uncontrolled movements. It is also called chorea. It is the dance in this disease. So that is the important link between GABA and the Huntington disease. Next function is the synthesis of the glutamine. So let's see the synthesis of the glutamine from glutamate. So glutamate is converted to the glutamine by the enzyme glutamine synthetase enzyme. Here ATP is utilized and there is incorporation of the ammonium ion. So there is formation of the glutamine. Now glutamine is broken down to liberate the glutamate by the enzyme glutaminase, that is the hydrolysis reaction. So there is a breakdown of the glutamine to form glutamate. Now the functions of the glutamine are: it helps in the protein synthesis, it traps the ammonia in the brain, it helps in the formation of the urinary ammonia and thus helps in the acid-base balance, especially the role of the kidney. It provides the nitrogen of the third and the ninth position and in the pyrimidine at the third position. It helps in the synthesis of the guanine and the cytosine. Glutamine helps in the detoxification reactions as well as production of the hexosamines. Now we see aspartate and asparagine, its synthesis and degradation. So oxaloacetate is the precursor. Through the transamination reaction, the aspartic acid is formed. Aspartic acid is converted to the asparagine by the enzyme asparagine synthetase, where glutamine provides mainly the nitrogen here instead of ammonium ion, and it is converted to the glutamate. Now asparagine is further converted back to the aspartic acid by the enzyme asparaginase. This reaction is quite similar to the conversion of glutamine to the glutamate. Okay, so there is synthesis of aspartic acid from the transamination reaction and the formation of asparagine from aspartate, and this is the degradation. Aspartate is degraded again to the oxaloacetic acid by the same transamination reversible reaction, and oxaloacetate enters into the glucogenic pathway. Now we see the function of both these two. So aspartic acids, it is the glucogenic amino acids. It is provides the nitrogen in the urea or helps in the urea cycle. The whole molecule of aspartate is incorporated to form pyrimidine, that is the first nitrogen, carbon 4, 5 and 6. It also helps in the synthesis of the first nitrogen in the purines and amino group at the 6th position. It also incorporates into the various protein for protein synthesis. Malate aspartate shuttle is also important in the electron transport chain, as well as it will transport the oxaloacetate. Now asparagine, it helps in the protein biosynthesis and also it ammonia in the brain. Now here the asparaginase is also helpful in the treatment of the cancer. Parenteral administration has been used in various malignancies, in particular the leukemias in adults, specifically in the lymphoblastic leukemia, because the leukemic cells, they are unable to synthesize the asparagine and they dependent on the blood level or dependent on the blood circulation for the asparagine. But if injected asparaginase, if we inject the asparaginase, it lowers the plasma level of the asparagine and thereby inhibiting their growth. So that is how, this is the reason behind using the asparaginase in the various cancerous conditions. These are my references for the presentations. Thank you everyone. Please like, share and subscribe to my channel. Click the bell button to get all the notifications. Thank you all.